Okay, so today we are going to continue the transcriptomics module. So last time we talked about mostly the platform, like microarray, nanosync, and RNA sequencing. So today we're going to focus on the analysis part. And we are still not done, so next lecture will still be about transcriptomics, but will be a different analysis of transcriptomics. So today we are going to focus specifically on differential expression, which is where we try to compare gene expression across multiple experimental conditions and try to identify genes that are up or down regulated. And then we can continue to analyze that information. Okay, so today we're going to, as I promised, so we're going to talk about the alignment of RNA sequencing directly to the transcriptome, not to the genome, and see what happens when we do that. We're going to talk about the many ways that people use to quantify gene expression, different units that people use, and how we can make sense of them. And then we're going to talk about how people model the read count that we get from RNA sequencing, what kind of statistical distribution can explain that data. And then we're going to talk about differential expression analysis from the perspective of statistics. Okay, so just to recap, we know that with RNA sequencing data, we have three main choices. If we are studying non-model organisms where we don't have any information at all, like no reference genome, no reference transcriptome, then we need to do assembly first. So that's the same concept as genomics. So we have to put all the reads from the RNA together, and hopefully we can reconstruct the whole transcript. And then we can use that database to align the individual reads. So basically, if we don't have a database, we have to create it from the assembly. But if we have a choice between the reference genome, which have both the exon and intron, and the transcriptome, which have only the exon, or the intron have been spliced, both can be used, and we will see the difference today. Okay, so to imagine, like when we have a reference reference genome, we have both the exon and the intron. But when we have a reference transcriptome, which is the process transcript, the mature RNA, the intron are already spliced. So the database will have all the exons connected to each other. And because when we do the RNA sequencing, we are mostly sequencing the mature RNA. So our read also do not have intron. So when we align our read to the transcriptome directly, so this doesn't, the software doesn't need to consider the possibility that you might have an intron that is missing in your read. So the alignment will be direct, will be ungapped. So we assume that the read and the transcript match as a whole. And there are special tools for this, Salmon and Callisto. So today we're going to talk about what these tools, what strategy, what analysis strategy, what algorithm that these tools use. And actually it's something that we have seen several times before. I think this is the third time. So when we want to do the alignment, but we want to do it quickly, the same trick that we talked before is to use a k-mer. So k-mer is the oligonucleotide, like only K nucleotide, that are part of your read or part of the transcript. So it's like a short segment from your read and from your transcript. So the idea is this. So we have the reference transcriptome, like so we know what are the possible RNA isoforms in our sample. Let's say we have 3 possibilities of them. So what we want to do is, instead of doing the full dynamic programming, like we have a read and we have the reference transcriptome, if we do the usual alignment, we have to do something like a dynamic programming between our reads and the transcript. But if we want to accelerate that process, what we are imagining is if the reference transcriptome, if the transcript isoform was the isoform that produced our read, if we look at the k-mer pattern on our reads, it should match to the k-mers on the original transcript, because that's the molecules that generate our read. So they're going to create a database. So the same double graph where we connect all the reads together. But in this case, instead of connecting all the reads, they're going to pre-process the RNA transcript. So we have a whole full-length RNA transcript. We're going to compute all the k-mer, like the short k-mer, from each region on the transcript. Each region produce a different k-mer. And they're going to create this graph that says that, okay, for my transcript in blue, it will start with this k-mer, followed by this k-mer, followed by this k-mer, followed by this k-mer, followed by this k-mer. So you're going to have a graph, a path on the graph that connects from the first k-mer all the way to the last k-mer. And these are generated because you know the full transcript, so you can generate this. If you have two isoforms that are similar, they're going to share the same k-mer, like the blue and the pink isoform. They are similar at the beginning, and then they differ at the end. So they're going to share the same k-mer all the way until the very end, where the pink transcript is shorter than the blue transcript. The green transcript is common with the other two in the beginning, and then there's some exon here that's unused. So the green isoform and the blue and the pink isoform is going to differ in the middle, where the green isoform has its own k-mer by connecting these two exons, which doesn't appear in the other two. So in the database, it will look like this. You have some k-mer connected to each other, and isoforms that are similar will share the same path throughout the graph until up to the point where they are different. Then you see different paths between the two. So what we are doing is, in our read, we can also produce the k-mers. So let's say our read comes from the 5' location on this isoform. So when we look at the k-mer from our read, it's going to match to the k-mer in the front of the reference transcriptome, of the reference transcript. So our read might have this k-mer, the one in black, this k-mer, this k-mer, this k-mer, this k-mer. So by knowing the k-mer profile on our read, compare it to the k-mer profile of our transcripts, we can tell directly that we can rule out the green transcript because the k-mer do not match. And now, the other thing that we can tell is this read cannot distinguish between the pink or the blue isoform because both of them have the same k-mer that are common to our read. But the fact that we don't need to do dynamic programming, we just need to figure out which of these isoforms, like which of these k-mer paths match to the one that we saw on the read. This algorithm is much faster than doing the full alignment. Because in this algorithm, we don't need to do any alignment at all. We just ask, is our k-mer from the read, like where is it on this graph? And we map the location, and from the location, we can rule out the isoform, and we can retain the possible isoform. So this is done very quickly. And this is to compare when this method first came out. You might be surprised that this technique sounds very simple, but people just thought about doing this only 10 years ago. So when this method first came out, they compared the time it takes for the software to analyze the RNA sequencing data compared to the traditional alignment-based approach. Like these are alignment-based. And these tools are based on k-mer. You can see how fast, the speed up that they get, like more than 100-fold speed up. So what they did with the amount of time that they gain, they gain many minutes, many hours by doing a new algorithm. What they are doing is bootstrapping. So we talked about bootstrapping before in the lecture for phylogenetics. When we have only one genome, when we build a phylogenetic tree, like we only get one tree, and we don't know how confident we are about each branch in the tree. It's the same for RNA sequencing. If you analyze all the reads together, you get one gene expression number per sample. You have no idea about how confident you are with that number. If a gene is found with 100 reads, how do you know that it's 100 plus or minus 5 or 100 plus minus 20? That is That gives you the same kind of average expression of 100, but in terms of statistics, in terms of analysis, knowing that it's 100 plus minus 5 versus knowing that it's 100 plus minus 20 make a lot of difference. And the only way you can tell whether your expression level is variable or not variable is you have to do bootstrapping. So basically, you have to subsample from maybe millions of reads that you get from each sample. Instead of analyzing everything together and get one gene expression, you can subsample, like taking some of the reads, not all of them, and then analyze it. You get one gene expression. You take a different set of reads, you get another gene expression. So by doing this replicate, bootstrap replicate, you not only get the average of gene expression by averaging, you also can compute the variance, the standard deviation for the gene expression. But imagine that back in the past where you have to do sequence alignment and you are spending 6 to 8 hours for a sample. If you want to do bootstrapping, we want to do maybe 10 bootstrap. And when we did the phylogenetic, we want to do 100 bootstrap. So you have to spend 600 hours just to get the variance from your RNA sequencing. It's not possible back in the past. But nowadays, now that we have new alignment techniques, we call it pseudoalignment because it's not a real alignment. It's a k-mer-based searching. But because we can do it so fast, we can do it 100 times, 20 times with no problem. So these tools not only introduce a faster way to analyze RNA sequencing data, they introduce the capability to estimate technical variance from the bootstrap into RNA sequencing data. And we're going to come back to this kind of additional power of the newer tools in a bit. Okay. But for now, let's talk about once we align or either pseudo-align RNA sequencing data, we're going to get read counts for each gene, how many reads map to that gene, how many reads map to that transcript. But we know that read count is going to be confounded by many other factors. If you sequence a lot, if you spend more money, you're going to get more read count because you sequence more. If you have a longer gene, your longer gene is going to produce more fragments, and you will get more read because you sequence more fragments from that gene. So people first came up with the notion of FPKM, fragment per kilobase of exon per million reads mapped. So it's basically spell out the normalization technique. You count the number of fragments, the number of reads. You divide it by two factors. First is the length of the gene. And in this case, because they want to make the numbers reasonable, we know that genes are usually in the order of thousands or 10,000. So they divided the transcript length by 1,000. So it says kilobase of exon. Not just exon, but kilobase of exon. So that the number is smaller. And then for the read count, usually we sequence in the order of millions, 10 millions, 100 millions. So instead of dividing by just the total read count, we divide also by 1 million to make the number small. As a human, we don't want to see many zeros or very small decimal digits. So we want to make it easy to recognize. So that's why the FPKM, when it was invented, is per kilobase of exon and then per million reads mapped. So it's not just the total read. They only consider the read that can be mapped to the DNA, to the genome or the transcriptome, because you can have contamination. You don't want to normalize your data by the contamination. So this is normalized to the number of reads that can be mapped, that can be aligned to the genome or to the transcriptome of your species. So people thought, at first they thought that this could be the answer. You normalize by the two things that affect the read count. The length of the gene and the throughput of your sequencing. But then people later found out that this is not really the same as if you think about when we analyze composition, right, we want to know how many bacteria are in the sample. Usually we thought of those as percentage. Right? These bacteria are present in 50% or 70% or 10%. So people found that we want to do something similar for gene expression. And SPKM, even though it's normalized by these two effects, is still not a composition. If you sum the SPKM for each sample, they're not going to sum to the same number. So people thought that why don't we convert SPKM to percentage? And that's why we have another unit called TPM, or transcript per million. So basically they take the SPKM, which already normalized for the gene length and the read count. They divide it by the total SPKM in each sample. So that now for every sample, your TPM is going to sum to 1,000,000 because you divided all the SPKM by the total and then multiplied it by 1,000,000. So this is like percentage, but instead of percentage, it's per million. And the good thing is now for every sample, your TPM is going to sum to the same number. So you can think of it as percent that you are more familiar about. And people found that when we do data visualization, or when we want to say whether sample 1 and sample 2, do they have similar gene expression profile, they found that TPM is better for calculating those metrics. So it just like by people trying out both ways and found that TPM is better for analysis, for visualization to be exact. So just to summarize, we know that we got the read count as the first unit of gene expression. We know that it's not normalized, but we don't want to discard it just yet, because when we want to do statistical analysis, the thing that we can model using statistics is the read count. So this unit, even though it's not normalized, we are not using it to analyze anything by our hand. It's the input data that all the tools want. So make sure when you analyze the data, don't throw the read count away. That's what all the statistical model wants. They want to start from there. But for visualization, usually we transform the data all the way to TPM. And then we can visualize as a scatter plot doing dimensional reduction, which we'll talk about maybe in 10 lectures. And we can also cluster the data, which samples have similar gene expression profile. We can do that with TPM as a unit. But for today, since we are going to talk about statistical analysis of gene expression data, we're going to focus on the read count, which is the raw data that we can apply statistics to. Okay. So I'm going to introduce a new distribution again. So last time I think we reviewed about Poisson distribution and binomial distribution before. But today we're going to see another distribution called negative binomial. And this is the distribution that people use to describe read count from sequencing data. So let's see why statisticians picked this distribution. So first, let's look at the real data. So the real RNA sequencing data are the one in scatter plot in gray. So people plot the relationship. So each dot is a gene. And for each gene, they plot the average expression level of that gene in the cells against the variance. So they plot the average expression against the variance. The reason they do this is because the Poisson distribution that we understand very well, we have seen several times, has a major restriction. If you describe anything with the Poisson distribution, the variance is always equal to the mean. It's just the way that Poisson distribution is defined. So if read count, which are integers like 1, 2, 3, 4, 5, people are tempted to model it with Poisson distribution. But if you use Poisson distribution, which implies that variance is equal to the mean, your data should follow the black line. This is where the variance and the mean are equal. And we can see clearly that in nature, read count that we get from genes is not following the black line. For highly expressed genes that have high expression level, the variance increase even more. It's much higher than the mean. So people found that no, we cannot use Poisson for RNA sequencing data. The second thing that people notice is if we try to imagine what kind of function follow the scatter plot of the data, right, it looks like maybe it's a parabola. It's a parabolic shape. Right? As you have higher and higher mean, the y-axis goes faster and faster upward. It's like a parabola. So people thought, what distribution whose variance is a parabolic function of the mean? And it turns out negative binomial has that property. So let's look at that. When they try to fit the negative binomial, it doesn't fit perfectly, right? But it captures the trend that the variance is growing faster than the mean, much better than Poisson. So let's see what the heck is this like distribution, negative binomial, what it is. So we know about binomial distribution. Right? So binomial, we have a event with certain success, probability of success, right? And we observe it many times. And we ask like how many success do we obtain? That's the vanilla binomial distribution. The negative binomial distribution focus on the failure. When we have both the success and failure, negative binomial is negative because we focus on the negative, focus on the failure. And the way it's defined is like this. So let's say we have some event or some trial. Like you might want to try something like throwing a coin or throwing a dice. You have certain probability of success, probability of getting the result that you want. And you want to keep doing this, like you want to keep throwing the dice or you want to keep rolling the coin many times. And your goal is to obtain certain number of success. Let's say you want to get 5 heads. You want to throw the coin, toss the coin many times until you get 5 heads. And then you stop. You want that 5 heads. But all along the way, until you get 5 heads, you might get many tails. You might get 1 tail, 2 tails, 3 tails, depending on your luck. So this distribution asks if we repeat this process with the coin tossing process until we get our heads or our successes, what's the probability that we will see a certain number of failure along the way? What's the probability that we see 1 failure along the way? What's the probability that we see 5 failure along the way? What's the probability that we see 10 failure along the way? And it turns out that can be calculated, not too difficult. So we know that there must be a certain number of toss, number of trial, and we know that some of them are failure, the rest are success. And the failure has probability of 1 minus p, and the success has probability of p. So it's just this k plus r minus 1 choose k, and then times the 1 minus p to the k, and then p to the r. So this is not that difficult. So people then, once we know the probability density of this distribution, we can calculate the mean. And the mean is p times r divided by 1 minus p. And you can think of this as if you have r success, like you have r successes, and you want to know what your expected number of failure, it's going to be proportional to the number of success. If you have more success, means you do more tosses, you're going to have more failure along the way. And the ratio is going to be equal to the odd ratio. P is the probability of success and 1 minus P is the probability of failure. So your failure is going to scale with the number of success with the proportion of the odd ratio. And then they can calculate the variance. And the variance, if you go to Wikipedia, it doesn't look like a parabolic function of the mean. That is until you kind of do some algebra to it. If you write this out as a sum of 2 fractions, it will look like mean variance is equal to mean plus mean square times a certain factor. So it looks like this distribution, whose description doesn't really match the read count at all. Like what happened in the cells when we do some probability of success and failure, like what does that mean inside the cell? But in terms of mathematics, this match what people need. We have some distribution that whose variance is a parabolic function of the mean, and that match to the observation. So up to this point, we might think that we are forcing some weird statistical distribution into our data. It doesn't explain anything in the RNA sequencing experiment. And that is until other people try to understand negative binomial distribution from a different angle. So if you go to Wikipedia, you will find that negative binomial distribution is equivalent to many things. There are many alternative ways to explain this distribution. This Bernoulli trials with the number of success and failure is the classic one, which gives this probability. But there are other ways that you can come up with negative binomial by going to different probability. So this one, hopefully you still remember enough calculus to appreciate this. So people found that we can write the negative binomial density as a combination, a mixture, right? Because we have a product. So this is a mixture of Poisson distribution. When you have a mixture, you have to have the distribution and you have to have the weight, right? How much of which fraction constitute of this Poisson. So basically, people found that negative binomial distribution can be written as a mixture of Poisson and gamma distribution. So what does it mean? So this means that if we think about gene expression, like we are still in the bulk gene expression, like so we extract DNA, RNA from all the cells like in the sample. We are not doing single cell yet. So we can imagine bulk gene expression that we are getting with the read count from RNA-seq. It's an average over many cells. We are analyzing thousands of cells at once. So imagine that if for each cell that we are analyzing, if the read count from each cell follow the Poisson distribution for each cell, and then each cell type will have a different population. In your sample, you might have a lot of fat cells, and then you might have some immune cells. So each cell type will have a different population. So if you imagine that maybe the proportion of the cells follow the gamma distribution. If we recall back to the phylogenetic lecture, we talked about gamma distribution as being very flexible. Gamma distribution can act like an exponential decay. Gamma distribution can act like a normal distribution. So basically, with this interpretation of negative binomial, we can think of it as the gene expression, the bulk gene expression that we are seeing, is the average of many cell types, and each cell type, its gene expression, its read count, actually follow Poisson distribution. But because each cell type have a different fraction, we have to multiply the fraction to the number read count to get the average. So this integration is the average of many cells in the sample. So if you think about this, then negative binomial makes some sense. We are analyzing the average, and we are seeing the distribution that is a combination of the Poisson, which is natural for counting events, and gamma, which can control the proportion of different cell types. So with this interpretation, it can make sense that we use negative binomial for RNA sequencing data. So hopefully that kind of when we first saw this distribution, like most students will be like, what? How do we allow this to explain experiment data in biology? But there are actually, in fact, there are ways to make sense of this distribution. Okay, so now we know that for last time we know that the intensity data from microarray and the number of molecules from nanosync, those can be treated as log normal distribution. And now today, we learned that for RNA sequencing read count, it can be captured with negative binomial distribution. So now we know the distribution. Now we can do the statistics. We can compare the across sample to identify genes that are differentially expressed. So let's start with the simplest one first. We're going to go back to the log normally distributed data from microarray or nanostring. So if we log transform those data, we're going to get normally distributed data. And that's the distribution that you are more familiar with. When we analyze sample between groups, like using t-test, these are when we assume that the data follow normal distribution. So you can imagine that for micro and nano string, once you do all the normalization, like remember that for microarray we correct for the background noise. For nano string, like we have both the negative control and the positive control to correct the expression level. Once we do the correction, the normalization, and we take the log transform of the data, then t-test is your tool. So basically it's very easy. Once you transform all the data, you can do this in Excel using t-test. And then after doing t-test, like we need to correct for multiple tests because we are perform as many tests as the number of genes. For each gene, we perform one t-test between control condition and the treatment condition. So we need to be careful of two things: which t-test to use and which method to correct the p-value. So just to recap, I'm pretty sure you are familiar with the flavors of t-test. Like we know that we can do one or two tails. If we have no hypothesis, we have no knowledge, we don't know if the gene will be up or down regulated, then we use two tails because we allow both up and down regulation. But if you have some hypothesis that your treatment, you expect it to knock out or knock down the gene, you expect it to be down regulated, then you can focus on that and you can do one tail analysis. Another flavor is whether you have pair or unpaired sample, or do you want to assume equal or unequal variance. So people, some students ask me this, like, should they use equal variance or unequal variance in their t-test? So if you have no knowledge at all, like no prior information, unequal is always safe. It's always safer because you allow the model to estimate the variance for both control and treatment. But if you know that, or somehow you know, or somehow you believe that the treatment that you are analyzing or the disease that you are looking at, it only changed the gene expression, maybe up by twofold or down by twofold, but it doesn't change the variation. Because some gene, like you can increase the expression, but as long as you don't disrupt the regulation, like the gene will still have the same variance. You just increase the expression by twofold. But sometimes if you disrupt the gene regulation, like if you disrupt the epigenetics, your gene might be more variable. Instead of having a stable expression, it might go up and down because you maybe knock out the regulatory factors that govern. So it depends on your assumption. You can select either, but usually it's safer to do unequal variance. Okay, and for the p-value correction, last time I think we already mentioned about how important this is, like when you have many tests from GWAS, if I remember correctly. At that time, we focused on the simplest technique, which is the Bonferroni method. So with Bonferroni method, we just divide the p-value cutoff by the number of tests. If we want to do a cutoff at 0.05 and we have 1,000 genes, we need to adjust our cutoff by 1,000-fold to get 0.0005. This is very easy because we can divide it by the number of tests. But in terms of theory, this is the method that has the least power. The least power being that you're going to reject, you're going to fail to reject the null hypothesis. So you're going to get less genes that pass the threshold. Okay. Another pitfall, not pitfall, but another limitation for Bonferroni is you are operating on the p-value. So the p-value, if we recall, is calculated based on the assumption that null hypothesis is true. But when we analyze the data, we are not focusing on the genes that do not change. The null hypothesis is that the genes do not change. when we analyze the data, we are actually focusing on the genes that are up or down regulated. And we want to be sure that those are correct. P-value doesn't really actually say anything about the correctness for those genes because it's calculated based on the null hypothesis, where we assume that there's no change. So we want something that can give us a better, a more direct control of how many false positives are we getting. And we have something called false discovery rate. So false discovery rate is the property of getting a false positive, so it's very direct. If you identify 100 genes as being differentially expressed, it passed your threshold for false discovery rate. Let's say your false discovery rate is 0.05, 5%. If you identify 100 genes as being significant at the false discovery rate of 0.05, it tells you directly that out of your 100 genes, you expect 5 of them to be false. So it's a direct determination that if you have 100 and your false discovery rate is 5%, you have 5 mistakes or 5 errors in your data, in your result. But false discovery rate is based on genes that are differentially expressed. It's a situation that we don't have any hypothesis. We don't have any model to calculate. So the good thing in statistics is that people found a way to control the false discovery rate, to control the p-value. So we are still applying the cutoff to the p-value, but we are not just applying a cutoff to the p-value. We are applying certain cutoff to the p-value so that we can say the cutoff about the SDR, about the false discovery. So I'm going to introduce 2 techniques, which is similar, very similar. The first one is Benjamin-Hochberg procedure. Some of you might have seen this before. Many of you probably have seen this before. So one thing that's different between this procedure and Bonferroni. So first, this procedure is going to control the false discovery rate, which is better than controlling the p-value. But there's nothing that you gain for free in mathematics or statistics. There's some assumption that you have to accept. But the good thing is these procedures are popular because they are varied under many assumptions. So you can have independent tests. But we know that in gene expression, the expression level of the gene are not independent. If 2 genes regulate each other, their expression level will be dependent. But Benjamin-Hochberg procedure allow for some correlation in your test. So even if your genes are not independent, you still allowed to use this type of procedure because it's still valid under certain correlation. So people can just take this as, okay, we can use it for gene expression, for more situation in biology. So the procedure is this, if you haven't done it before. So we have the p-value from our genes, so 1 p-value for 1 gene. What we want to do is we're going to first pick our false discovery rate cutoff, let's say 5%. We're going to sort the p-value from low to high. We know that the lower p-value is always better than high p-value. So we're going to sort our genes based on the p-value from low to high, so from best to worst. And we are going to try to see if we can reject the null hypothesis for each gene, going from the best one to the worst one. And the way that we are doing it is, for each gene, we have the rank, like the best gene will be the first rank, the second best will be the second rank. So the subscript I here is the rank of the gene. This is after the sorting. So for the first gene, I being 1, we're going to compare the p-value for the best gene to get to the 0.05, which is our SDR cutoff, multiplied by I, which is 1, divided by the number of tests, the number of genes. So you may notice that when I is equal to 1, this is exactly Bonferroni. Because Bonferroni, you divide your p-value cutoff, in this case SDR cutoff, by the number of tests, the number of genes. So for the best gene, you are essentially doing Bonferroni. That's the best you can do. So Bonferroni, you still need you're eating exactly Bonferroni, but doing it only for the best gene, the gene with the lowest p-value. But when you look at the second gene, the second best gene, your cutoff is twice, like it's times 2, like it's twice the Bonferroni cutoff. So you can see that this test allow more gene to pass. The first gene still follow the Bonferroni cutoff, but the second gene is more relaxed. Like the cutoff is now twice as high. It's easier for the gene to pass. And for the third gene, like the cutoff is 3 times as high. So the third gene is easier to pass. So we'll keep doing this. Like if the first gene pass, we'll go to the second gene. If the second gene still pass this, we'll go to the third gene. All the way until at a certain point, it no longer pass, like the p-value is greater than this. Then we stop. So basically we are going to, we'll see some example to help you understand this process. So we are sorting the gene from best to worst, and we are going to try to reject the null hypothesis for each gene one at a time from best to worst. And as long as we can keep rejecting the null hypothesis, we will keep going. But once we fail to reject the null hypothesis, we stop. We don't go further. So let me introduce a related technique first, and then we'll go see the example. So another technique, which is very related, the second person just changed the name from Hochberg to Yekutieri. So these two tests are very similar. I'm not going to burden you with the details, but it's the same procedure. The only difference is this factor. For Benjamin Hochberg, it's 0.05, which is a cutoff, times the length of the gene divided by the number of tests. For this Yekutieri variation, there will be another factor that multiplies here. The difference between the two is for Yekutieri is more generalized than the Hochberg version. So with the Hochberg version, it works on independent tests. It works on some positively correlated tests, but there are some situations where you shouldn't use it. And some of those situations, you can still use this procedure. So this procedure is more stringent, slightly more stringent, but it's more general. You can use it on more situations in statistics. So let's see how these three differ. First, in terms of cutoff. So let's say we sort the gene from the smallest p-value to the largest p-value. Bonferroni is bad because you are applying the same cutoff for every gene. Let's say 0.05, and you have 100 tests. You're going to apply 0.005 cutoff to every gene. So this is the most stringent. For Benjamin Hochberg, because the cutoff keeps getting larger, from 0.05, which is equivalent to Bonferroni, to 0.01 times 2, 0.015 times 3, 0.002 times 4. So the cutoff keeps getting larger. So it's easier for the gene to pass. For Benjamin Yekutieri version, the factor here is slightly more stringent. So instead of 0.001, it's slightly less than 0.01. Instead of 0.015, it's slightly less than 0.015. But it's still larger than Bonferroni. So Bonferroni has the least power because the cutoffs are so low. For Benjamin Yekutieri, the cutoffs are slightly higher than Bonferroni. For Benjamin Hochberg, the cutoffs are even higher. So you have more genes that pass. So let's see this example where I apply to some engineer p-value. So these are the p-value from 0.001 to 0.06014. If we apply Bonferroni, 0.005, so the first gene will pass, the second will pass, and the rest will fail. But if we apply Benjamin Hochberg, 0.05, the first gene pass, 0.01, oh, the second gene pass, 0.015, oh, the third gene also pass, 0.002, the fourth gene still pass, until the last gene, 0.025, this is higher. So this fails. For the Jekyll variant, because the cutoff is smaller than the Hochberg variant, you have less gene passing the cutoff threshold. So my recommendation, if you want to be a good statistician, you will use the Jekyll version. But if you want to kind of close one of your eye, you can use Benjamin Hochberg. And if you are too lazy, you can use 140. But usually that comes with a lot of lost genes because the cutoff is very low. So the trade-off usually is to do the Benjamin Hochberg procedure. Okay. So that's for the microarray and nanosync. The analysis is easy because we can use a t-test. But for RNA sequencing, we can no longer do that. The data has to be fitted to the negative binomial, and we cannot do that by hand. There is no tool for that. So we're going to rely on some bioinformatics software that people have developed. And I picked two software here for us to look at today because they operate on different assumptions. So the first one, this one you might have seen a lot in literature, DE-seq2. This is developed before we have the k-mer-based technique. So it assumes that you are doing regular genome alignment. You get the read count, you don't have any bootstrap. You only have one number per sample. So inside the model, it's going to try to estimate everything for you because you don't have the bootstrap. So it's going to model the read count that you get from the alignment as a negative binomial. So the negative binomial, there is a mean and the variance. And we know that the variance is a function of the mean, like the mean plus some factor times the mean square. So they're going to use these properties of negative binomial. So the DE-seq2 is going to have to estimate for each gene and each sample. So gene I in sample J is going to have to estimate the average expression for all the genes in all the samples. And they're going to break the average expression down into two effects. One is based on which sample that you are analyzing. Different conditions give you different average expression. And also your sequencing, the technical bias. So let's start with the simple one first. The sequencing effect basically are the things that we already talked about. So the sequencing depth, it affects the number of reads that you have. The GC content of the read can bias the amplification, the PCR process. And the length of the gene, we know that it can produce longer gene, produce more reads. So basically it's going to apply those principles inside the tool. But it's a principle that we already understood. So this is the sequencing effect that can change the average expression. What about the sample effect? So the sample effect means that which condition, like which experimental condition, maybe you have a different dose, different dosage of the drug. Maybe it's a time, like 6 hours after treatment, 12 hours after treatment. Maybe it's the age of your sample. Animal or patient with different age can have different gene expression. So these are all parameters that the software or any model have to use in order to model the average expression, which condition, and other factors. So the way that DE-seq2 takes your experimental design, like control treatment, or other confounding factor like the age of the sample, time after treatment, dosage, for example. The way that is incorporating this into gene expression model is via this kind of linear effect model. It's going to assume that the log fold change between two samples. So the difference, basically, the difference in gene expression between two samples on the log scale is going to be modeled as a linear function. Up to this point, you might start to realize that in bioinformatics, when we don't know anything, we use a linear model. because that's the model that we can solve very easily. But we have to define all the parameters, like which effects goes inside the model. So in this case, it's going the DSeq2 imagine that the fold change in gene expression on the log scale between any two samples is going to be a combination of the difference in experimental condition between the two samples. If they have the same condition, then there is no effect. If they are different condition, then there is an effect from the condition. If the two samples receive different dosage or it's different time after treatment, then those will be changing the fold shape, affecting the fold shape. So basically, this is how they model. The beta is the effect size, like the coefficient, and the x will be the indicator, whether it's a control or treatment. X may be the age, maybe the dosage. So those are the actual difference in the design, the experiment parameters. And the effect size, which is unknown. Okay. So to give you some example, so we might have some experiment. We might have two treatment, and in some sample we might not treat anything. Like it's all 0, no treatment. In some sample we treat it with the first drug or the first molecule. So we have 1 here in 0. In some sample we treat it with both compound, both drugs. So we have 1 in both. So this matrix you just create based on your experiment. Which treatment you use, 0 and 1. If it's the age of the sample, you put in the age. If it's a dosage, then you put in the dose. And the DSeq model is going to assume that the change in gene expression is going to be maybe a certain constant plus 1.09 times whether drug 1 was treated plus 1.97 whether drug 2 was treated, and maybe 0.82 times the interaction between the drugs. So once you define all your experiment parameters and the confounding factor, they're going to be incorporated into the gene expression model as a linear function, as a linear combination. Okay. So from this kind of linear effect model, linear combination of the design parameters or the confounding factor, multiplied with the effect size, how do we perform differential expression from this model? So this, I would name this as a differential expression as a test of effect size. So we are incorporating all the design parameters like treatment, control, dosage. If those factors do not have any impact on gene expression, the beta should be 0. If the parameter do not affect the gene expression at all, like beta should be 0 because you don't want any effect from that parameter. So the test that they are doing here, once they fit the observed expression data to this linear effect model, the DSeq software is going to ask, is the beta for treatment, or is the beta for dosage, or is the beta for the time after treatment? Are those beta significantly different from 0? If it's yes, the beta is not 0, then that's your effect. Your drug or your treatment has some effect. It can be positive or negative. As long as it's not 0, you have some effect. It might increase or decrease your gene expression. But if your beta looks like it's 0, it's very small, then you can say that maybe that parameter doesn't have any effect on your experiment, on your gene expression. So this is done with a wall test. So it's a kind of a statistical test that people use for effect size. And the assumption behind wall test is that your effect size, if you estimate the effect size multiple times, your effect size divided by the standard error, this is going to be normally distributed. So underneath is like a t-test, but there's a t-test that adapted for testing the coefficient or the effect size in the model, and they call it wall test. So this is one way that we can identify differentially expressed gene. So whether that gene has effect from the experimental condition, then those are the genes that change. For genes where the betas are 0, those are the genes that do not change, for example. So this is one approach. Okay. So let's go back to in the DE-seq model. Like we just mentioned about the average expression being governed by two effects. One from the technical effect, the sequencing data, the PCR amplification. The other one is based on your sample, your treatment condition. Another thing that the DE-seq tool has to estimate is the variance. So once the tools can estimate the mean, the mu here, it only knows some component of the variance. But it still have to estimate the factor here that multiply to the mean square. This gene specific effect on the variance. Because different genes will have different expression variance. So their assumption is, so we can be naive. For each gene, we can estimate the variance across the replicate. Let's say we have 3 replicates. For each gene, we can estimate the variance just by calculating the variance for those 3 numbers. But that might give you an incorrect estimate, because usually you don't have many replicates. You're probably not going to do like 20 replicates. So you don't have a good estimate for the variance if you do it for each gene. So we want to try combining information across genes. Can we look at genes with similar expression level? If we assume that these genes have similar variance, then we can average them together and get a more robust estimate for the variance. So what I mean by that is, let's go back to this when we first saw the expression data. We saw that as the genes have higher and higher expression level, like the average expression level, we get higher and higher variance. And we see that the variance is very consistent, like we don't see outliers. We see it's very consistent. So we can assume that, these genes that have the same expression level, they have the same variance. So if we combine information across these genes, we get a better estimate for the variance. Versus if we calculate the variance for every gene, we get very scattered, very diverse estimate. So this is the principle that the DE-seq tool used to estimate the variance. So if we plot, again, the average expression level, the y-axis now is not really the variance, but is a modified version of the variance. It's called dispersion. It's a variance subtracted by mean divided by mean square. So this convention is to remove the effect of the mean from the variance. And in this case, it's perfect for negative binomial. Because for negative binomial, we know that the variance is the mean plus some factor times mean square. So when we subtract the mean, like we move the mean on this side, and we divide it by the mean square, essentially we are plotting the graph for the gene-specific variance. So that's why they plot it this way. The y-axis here is the gene-specific factor on the variance that we want to estimate. So in this case, they're going to estimate, and by assuming that genes with similar average expression have the same variance, or basically have the same dispersion. So they are detecting this red line and the blue dots here that explain the majority of the gene. They are not going to be considering all these outliers. They're going to trust that if the gene really have similar variance, the trend should be in the middle. It should explain the majority. So this is a kind of regression. Like when you do regression, you want to fit a trend to your data. When you have outliers, data that are very low or very high, sometimes these can pull your average away from the center. So this kind of regression, sometimes called robust regression or sometimes called local regression, is a curve fitting techniques where we want to trust the median or the majority over the outliers. So this is the approach that D6 use. I think I will skip this. Maybe it's too deep for us. But in D6, they are not only trying to ignore the outliers, they are also going to use the principle that we learned earlier, the Bayesian idea, to improve the estimate even further. But let's keep it at this to not make it too complicated. But what we can see is the effect of doing using the Bayesian idea to improve the estimate for the variance. So if we trust the variance from the graph as it is, these two genes are going to have the same p-value, the same fold change. But if you look at the data, you can see that for the first gene, between the two conditions, the expression level shift and the variance are small. You can see that the distribution are different. But the second gene, the average expression also go up. But in this other condition, the expression level is very variable. The variance is very high. So if you estimate the fold change directly without considering this change in variance, these two genes give out the same fold change. But once D6 using this kind of a technique to look at the variance of other genes to make the adjustment, it realized that, wait, this second gene, the variance are really high. The fold change might not be trustworthy. So it reduces the significance for this second gene further. So this technique can allow you to distinguish better between genes that are consistent and genes that are variable. So let's just focus on the effect and not worry about too much about the actual mathematics. But basically they are incorporating this technique to improve the confidence of your fold change estimate and your statistical analysis. Okay. So that's the first tool, D6. It assumes that you don't have bootstrap data. It is from the era where you have only the alignment result. But since 10 years ago, we have new techniques, pseudoalignment, that is very fast, and we can do bootstrapping to estimate the technical variance. So the same people, the same risk scientists who develop those techniques also develop a new way to do differential expression. So they assume that in your data, your read count is going to be affected by two types of variance. One is biological. So if you go from one mouse to another, go from one plant to another, one patient to another, even if they have the same disease, there's going to be some biological variation. Different person is not the same. And then there's going to be what they call inferential or technical variance, which is caused by your laboratory protocol. But because we have the bootstrapping data from the pseudo-alignment, we already can estimate the technical problem. We can estimate the technical variance. We know that if we do sequencing multiple times, we simulate sequencing multiple times in bootstrapping, we know how varied would our gene expression be. And that captures the technical variance. If you do the same experiment from the same biological sample multiple times, what kind of variation you are getting? You are estimating that from the data. So they are arguing that now the only thing that needs to be estimated is the biological variance. And by separating out the biological variance and the technical variance, they are assuming that the variance are normally distributed. I forgot their argument exactly, but they, instead of using the negative binomial model as it is, they are still using the read count, but instead of assuming that the variance distributed according to the negative binomial. They argue that if we can separate out the biological noise, so this the orange is the biological noise, and the green is the technical noise. They argue that if we can separate out the two, both should be normally distributed. Because in biology, the expression level change from cell to cell. If once we take the transform the data, it should be normally distributed. Also, the technical variance, once we transform the data properly, it should be normally distributed. So they're assuming that the observation that we are getting, the capital D here is the read count, is going to be affected by, again, like X and beta. So X would be the experimental design, the treatment control, the confounding factor. Beta would be the effect size. So this will determine the average of your expression. But the variance is going to be normally distributed biological variance, which is unknown, has to be estimated by the model. And the technical variance, which is also normally distributed, but is known from the bootstrapping. So basically, they reduce the data to just estimating the biological noise. And this is easier because the noise is smaller than before. And now, I'm going to introduce another way to do differential equation, because these tools, you can see that it also uses the same linear effect model. So you can still use wall test to check if the beta is significantly different from zero. You can still do that with this technique. But these tools provide another way to look at gene expression, differential expression. So they're going to say that you might have different hypotheses, and the tools is going to fit the beta, the effect size, under different hypotheses, and then they will compare which hypothesis and the produced beta, which one fit the observed read count better. So this comes back to the likelihood ratio test that we saw several times already. So let's say you have three hypotheses. You have 4 samples, 2 conditions, S and L being 2 conditions, and you have batch. Like you might send S1 and L1 sequence first. Like after 2 months, you might send S2 and L2 to the same company. So your hypothesis, number 1, is that there is no treatment effect. No effect at all. All the 4 samples should be the same. Your second hypothesis is that your treatment, S and L, has some effect. So you think S1 and S2 are similar, and L1 and L2 are similar, but S and L are different. This is your second hypothesis. Your third hypothesis might be the treatment has some effects, but also you have some batch effect. When you send S1 and L1 together for sequencing, and when you send S2 and L2 together for sequencing 2 months apart, there might be some batch effect from the company that you don't know. So this might be your hypothesis. So the model is going to fit the beta together with these different X. So once you have the hypothesis, you're going to change your X, the X value. Whether there is no parameter at all, 1 parameter, which is the treatment, and 2 parameters, which is the treatment and the batch. The model is going to fit the beta. So for the no treatment, there is no beta to fit. For hypothesis 2, it's going to fit 1 beta for the treatment effect. For hypothesis 3, it's going to fit 2 beta, 1 for the treatment effect, 1 for the batch effect. So now you have 3 hypotheses with different beta, and each will have different likelihood, like how well it fits to your exploration data. If your gene is expressing at the same level across 4 samples, your hypothesis 1, your model 1, might fit it better. But if your gene is different between S and L, your hypothesis 2 might fit the data better. But if your gene not only differ between S and L, but also differ between batch, your third hypothesis might give you the best fit. So you can compare the likelihood using the principle that we talked about before to identify which hypothesis to accept. And in this case, the degree of freedom, like the first hypothesis, has no degree of freedom. There is no effect. The second hypothesis has 1 degree of freedom. There is the treatment effect. The third hypothesis has 2 degrees of freedom. So you have more complex model. And you can try to reject the complex model or reject the simple model by looking at the likelihood, as we have seen before. So just to show you that with this new approach where we can do bootstrapping and we can have a better handle of the technical variance, there is some implication when you analyze the data. So this is the paper that came out with the new tools. They compare their approach with other differential expression software. So the seq tool is somewhere here in blue. What they are showing is when you analyze your differential expression, you want to apply a certain fault discovery rate cutoff, FDR cutoff. So you are hoping that if you, let's say you apply a cutoff at 10%, and you get 100 differentially expressed genes, you are hoping that no more than 10 genes would be false, because this is your fault discovery rate. But when this software, this algorithm, analyze the data, fit all the data, perform the test, even if you set the cutoff at 10%, you're not guaranteed to get 10% because you set the cutoff, but the tools have to fit the data. There can be errors. There can be something that deviates from the assumption of the model. So you don't really get the 10%. So the y-axis here is the fault discovery rate. So this paper shows that when you use other tools that have no idea about the bootstrap, no technical variance estimate, you want 10% fault discovery rate, but you are getting 25%, 35%, 50%, 60% from the tools because they lack that bootstrapping information, and both at the gene level and at the transcript level. So this is the benefit of doing the new pipeline, having the bootstrap information and use that information to perform the statistical analysis. So this is in terms of the accuracy, like how much can we trust the gene, that the differential expressed gene that we got from the software. So I would recommend that you use these tools in your analysis. What about in terms of how many genes you are getting? The accuracy is how much can you trust the gene you are getting. It doesn't tell you how many genes you are getting. You might have a very accurate differential expression, but it only produces 10 genes. Versus you might have a less accurate gene differential expression result, but it gives you 100 genes. Sometimes you want more genes so that you can make hypotheses in terms of biology. So this graph shows you the number of genes that you are getting from each software, at the gene level and the transcript level. So the black line is the black line and the yellow line are the best tools that we saw in terms of the accuracy. In terms of the number of genes, they're still better. So they don't have kind of pitfall in their approach. And what I want to highlight is the seq tool, which is the state of the art for a while, the one in blue. It performed okay at the gene level. The blue graph here is not that far off from the black and the yellow. But if you want to analyze at the transcript level, you want to analyze an individual isoform, knowing the variance from the technical replicate gives you much better result compared to other tools. But if your goal is to study gene level, you don't lose much whether you use the new software, sleuth, or you use the seq tool. But if your analysis will be at the transcript level, at the isoform level, at least from this result, it suggests that you should try at least the new tools. Okay. So just to summarize what we talked about today, for microarray and nanostring data, these are not normally distributed. So once you take the log transform, you can perform t-test and then do the p-value correction. You can do this in Excel or any software. Nanosync data, because it's a molecular count, you can also think of it as a kind of a read count, except the numbers are high. It can also be analyzed with D-seq too. You can pass the read molecule counts from nanosync into D-seq too. It still works using the same model. But for RNA sequencing, it's a read count. You cannot do t-test. Don't try to log transform it and do t-test. If you align the data to the genome, which sometimes you want to do because you want to discover new isoform, new splice junction, the trade-off is you are not going to get bootstrapping. You're not going to get the technical variance. So your only choice will be to pass it through D-seq too. And this is the pipeline that in the assignment number 3. I'm asking you to do the alignment to the genome and then pass the result to D-seq too. But alternatively, if you align your data to the transcriptome using the k-mer techniques, which is much faster, and you also get the bootstrapping, you can put the data through either D-seq2 or sleuth, because now you have the bootstrap. You can decide not to use it, or you can decide to use it. You can pass it through both software. In terms of the actual practice, there might be some additional steps that you need to perform in order to make the data from one tool to be compatible with another. But luckily for you, if you search, there are people who create like a script, either in R or Python or any language, that can convert output from the alignment, like whether to genome transcriptome, to different software. So you can plug any output to any software. But if your analysis want to focus on the transcript level, at least from the evidence so far, you should try this software. Okay, that's everything we want to cover for today. So the next lecture, we're going to continue from this point. So now we know the techniques for identifying differentially expressed genes. So the next step would be to try to annotate those genes with biological function and try to interpret or make hypotheses about which function or which pathway are upregulated or downregulated. So that will be our next and final step for transcriptomics. Okay.